Hello everyone, welcome to today's session. Today we are going to see class 12 biology chapter 5 molecular basis of inheritance. In that we are going to see translation. Let's start with the topic. Now we are going to study translation. So the translation, it is the final major step in the process of gene expression. And we already know that the information stored in the DNA is first copied into mRNA through transcription process. So all these things we saw in our previous videos. So now this information present in the mRNA, it is used to produce a polypeptide or protein. Polypeptide or protein. So this process is called translation. And translation, yes, translation is the polymerization of amino acids to form polypeptide chain. So the sequence or order in which these amino acids are arranged in the polypeptide, it is determined by the sequence of bases present in the mRNA. Sequence of bases present in the mRNA. So the amino acids, they are joined together by a special chemical bond, called as the peptide bond. And forming the peptide bond, it requires some energy. It requires energy. So this before the protein synthesis begins, so the amino acid first should be activated and attached to the specific tRNA. So this process we called as the charging of tRNA or amino acylation of tRNA. So the entire translation process, it can be like broadly divided into three stages. So first stages is initiation and then elongation and termination. So let's see this one, activation of amino acids, yes, charging of tRNA. So what happens here? So this one, amino acid plus ATP. So before an amino acid can be like incorporated into a growing polypeptide chain, it must first be activated. So this requires some energy in the form of ATP. So the amino acid is first activated using ATP and then it is attached to the specific or cognate tRNA, attached or linked to specific or cognate tRNA. So once it is attached to the tRNA, it is called as charged tRNA and also known as amino acyl tRNA. So this process can be like understood as amino acid plus ATP, right? Activated amino acid, and it is linked to the cognate tRNA and the charged tRNA or amino acyl tRNA. So this charged tRNA, now it is ready to participate in the translation process. Now here are some exam tips for you. So if two charged tRNAs are brought close to each other, the amino acid attached to them are positioned close enough for a peptide bond to form. So the ribosome then it acts as a catalyst to increase the rate of the reaction. Next, let us see the ribosome, the protein factory. So before going into this, we should understand the structure and function of ribosome. Now we know that amino acids has been activated and it is attached to the tRNA. And where does protein synthesis actually occur? Anyone know? Answer is very simple. It occurs in the ribosome. So the ribosome is the cellular factory which is responsible for protein synthesis and consists of structural RNA and about 80 different proteins. So when inactive, your ribosome exists as two separate units. One is the smaller unit and another is the larger unit. They are subunits. So the small subunit, it binds to the mRNA. Here you can able to see a small subunit. It binds to the mRNA. Yes. And once it encounters with this mRNA, the process of translation begins. So the process of translation begins. So this is the mRNA strand and this is the small subunit of ribosome. And this one is the large subunit. It contains two sides where the tRNA is carrying the successive amino acids can bind here. And these sites position the amino acids which are close enough to each other for the formation of a peptide bond. So you can able to see the smaller subunit here and also a larger subunit, right? So the mRNA now passes through the ribosome. This mRNA passes through the ribosome and the ribosome reads its codon one after another. So this is a very important thing for your CBSE examination. So here are some exam tips. So the ribosome itself is the catalyst. So in bacteria, 23S rRNA, it acts as an enzyme or we can say as a ribosome. So next we are going to see the mRNA translational unit. So let's see the organization of mRNA. Organization of mRNA. So now let us understand the structure of mRNA that is used for translation process. So you should understand what is the translational unit. So a translational unit is a sequence of mRNA or a stretch of mRNA that is present between the start codon and the stop codon, and it codes for a polypeptide. It codes for a polypeptide. So the translation begins at the start codon. So the mRNA, it also contains some additional sequences that are not translated. These are called as the untranslated regions or UTRs, where you can able to see. So these are the untranslated regions or UTR. So there are two types in this. Again, at the 5' end, you can see 5' UTR. So it is present before the start codon. That is, what is the start codon? AUG. And then another one is present at the 3' end. This is 3' UTR. So it is present after the stop codon. So these regions are not translated into amino acids, but they are very important for the translation process. So you can able to see. So the mRNA stand, it can be represented as this 5' 3'. You can able to see the UTR here, and this is a start codon, and these are some coding regions. So what happens? See the ribosomes. It begins reading at the AUG start codon and it continues reading the mRNA codon by codon until it reaches a stop codon. So it means that yes, so many codons are there, right? And when it reaches the stop codon, it stops. And we know that already stop codons. What are the stop codons? UAA, UAG and UGA. So the region between the start and the stop codon is the region that is translated into a polypeptide. So this region is translated into a polypeptide. So next, we are going to see 3 steps of translation. So translation occurs in 3 major steps. So what are they? One is the initiation, elongation and the termination. So what happens in initiation? First, the ribosome binds to the mRNA. The ribosomes, this is the ribosome. So this is the smaller subunit and this is the larger subunit. It binds to the mRNA. And what happens? So the first, the ribosome binds to the mRNA. Where it binds? At the start codon AUG. At the start codon AUG. And then what happens? The AUG codon, it is recognized by the initiator tRNA. So the initiator tRNA, yeah, recognizes it, and this marks the beginning of protein synthesis. And next process is the elongation process. So after initiation, what happens? The process enters the elongation phase. So this is the elongation phase. So the charged tRNA, it sequentially enter the ribosome. Say how it is entering? It is enters the ribosomes. And then what happens? The anticodon of each tRNA, these are the anticodon of tRNA, it pairs with the complementary codon of the mRNA. These are the complementary codon. You can able to see here UA, UA, yeah, UA, yes. So the anticodon of tRNA pairs with the complementary codon of the mRNA. And then the ribosome, it moves along the mRNA. It moves like this. Yes, moves along the mRNA. And then what happens from one codon to the next codon? And as it moves, amino acids are added one by one. Amino acids are added one by one to the growing polypeptide chain. And then amino acids, again they are joined by peptide bonds. These amino acids, they are joined by the peptide bonds. So what happens here? codons on mRNA, and then anticodons are there on the tRNA. So this ensures the correct amino acids are added in the correct sequence. And finally, termination process. So the ribosome, it reaches this ribosome, it reaches stop codon. So a stop codon does not code for any amino acid, that we know already. So there is no tRNA for a stop codon, no tRNA for a stop codon. So instead what happens? A release factor binds to the stop codon, and this terminates the translation and also causes the completed polypeptide chain to be released. So you can see this diagram. Yes. So it shows the ribosome, how it is moving along the mRNA. Again, this charged tRNA enter one after another, and it recognizes the appropriate codons through the complementary base pairings here. You can see, and then the amino acids are joined together and they form an increasingly longer polypeptide chain. So at the stop codon, the release factor terminates the process and the completed polypeptide is released. So we saw the initiation process, elongation process, and the termination process. Yeah. Let's move on to the last part. Let's have a quick revision. So translation is the polymerization of amino acids to form peptide. And then a peptide bond joins the adjacent amino acids. And before translation, amino acids are activated and it is attached to the specific tRNAs. And this we called as the charging or amino acylation of tRNA. And then the ribosome is a cellular site of protein synthesis. It consists of larger subunit and smaller subunit. And then in bacteria, 23S rRNA acts as a ribosome, yes, and it catalyzes the peptide bond formation. And the translational unit of mRNA, it extends from the start codon AUG to the stop codon. And then the mRNA also contains 5-3-UTR, which are not translated but help in the efficient translation process. And what are the three stages? Initiation, elongation, and termination. In initiation, ribosome binds mRNA and starts at AUG. So in elongation, this charged tRNAs that bring amino acids and peptide bonds and form as the ribosomes moves codon by codon. And the termination part, a release factor, it recognizes the stop codon and the completed polypeptide is released. So from the whole thing, what we should understand is DNA is transcribed to mRNA. mRNA is translated to polypeptide, and the polypeptide, it results in a functional protein. So remember all these things. So what happens in this whole video? CRMIET. We can say CRMIET. C means charging of tRNA. R we can say as a ribosome, and M is mRNA translational unit, and the first process is initiation, E is elongation, T is termination. So we have concluded this part. Watch it fully, don't miss it. Yes, we'll see you in the next video. Thank you.